Hi everyone, it's my great honor to present my project here. Today I'm going to share with you some updates in structure-function coupling by combining fMRI and MEG data, as you can see from the title. This may not sound completely new, but I'll try to convince you why it's still fresh and deserves a lot of further study today. Before going into the main results, I first want to give you a very fast impression of the subject today, that is structure-function coupling. In simplest terms, we look for connections between the structure and functional brain networks. And of course, this is a great simplification. So to show how it expands from this, I will share with you some recent progress from our lab and explain how we landed on this fMRI MEG project today. So it all started by noticing that structure function representations are imperfectly matched across the cortex, making it critical to study the heterogeneity of the coupling across different brain regions. We have made use of many cortical annotations with concrete biological meanings. For example, structure function coupling follows the unimodal transmodal hierarchy at the macro scale and shaped by different neurotransmitters and gene expressions at a much finer scale. And when static annotations are not enough, we've also integrated many emergent and very promising methods to better describe this relationship. But what I'm going to show you today is not one of these more complex methods. Instead, it was simply from exploiting the function or the temporal part of this relationship. We've tried many methods, and without surprise, we landed on MEG as a measure of faster functional activity. I'm sure I don't need to explain how MEG excels fMRI in terms of temporal resolution and as a more direct measure of the electrical activity through magnetic fields. So Dr. Galia Shafi recently graduated from our lab, really did a lot of fundamental work comparing BOLD and MEG networks and make MEG data part of our analysis workflow. We realized nothing can make it more clear than just a direct comparison of structure-function coupling using both BOLD and MEG as the functional component. And what I'm presenting today is a back-to-back comparison of structure-function coupling using fMRI and MEG, focusing on the importance of using structural connectivity as a reference and how communication models arising from the structural network can improve and help explain what is going on. We're also going to try explain the importance with, apparently, cortical hierarchy and probably more fundamentally, cytoarchitectural annotations. So using data from HCP young adults, we derived diffusion MRI-based weighted structure connectivity and resting state functional MRI and MEG connectivity matrices. Then we estimated group consensus matrices from each of them. And for the structure part, we introduced communication-informed structural predictors to model the polysynaptic connections on the structural connectome. These are representative and commonly used ones ranging from routing to diffusion, capturing more potential dynamics that could happen on the structure. The effect of including them would be very clear once we get to the results. For the functional part, MEG functional connectivity matrices were derived for each canonical band using amplitude envelope correlation measure. And this is how they finally look like. So the basic method for the coupling is a simple multilinear regression where we use structure to predict function and using the goodness of fit to measure the extent of coupling. There have been several, but not a lot, previous work trying to link structure connectivity with faster dynamics like MEG and EEG. This is most likely because trimodal dataset is very rare, especially regarding MEG. Luckily, things have improved quite a bit, and there has been also more progress and consensus regarding MEG preprocessing and analysis. And among this previous work, many aim to construct mechanistic models of the activity, while here we take a very different approach by directly describing the statistical correlation. The multiple regression model, albeit simple, has helped us to identify many fundamental, robust, and explainable conclusions, such as the cortical heterogeneity of the structure-function relationship. We hope really to make it a simple and clear idea of how structure-function coupling look like between BOLD and MEG with our updated understanding of structure-function coupling. Okay, now it's time to put them together and start with the results. So we start by considering the regression model under two iterations. First, whether we assume cortical heterogeneity or not. We call it global versus local. And secondly, whether we only use the structural connectivity or an extended set of communication-based predictors. We call it univariate versus multivariate. And this gives us four scenarios progressively, and I'll show you how these factors influence our results. So first, we have the global univariate model giving one coupling value across the cortex, and it is clear that BOLD has the highest coupling compared to any of the main frequency bands. But wait, when we add the cortical heterogeneity by modeling each node independently, it is clear that a lot were hidden if only a single global value were considered. And what's more, when we add the communication models to predictors, it displayed a vastly different coupling pattern. It seems it's not like MEG bands are better coupling as in the first figure. Here, many slower bands are much better coupled to the structural connectivity than BOLD-derived network. This really tells us communication models potentially captures more dynamic processes on the structural network and are useful for examining the faster dynamics of MEG. This is also shown by the differential contribution of communication predictors in the regression model. Finally, we have the local multivariate version with both node level coupling and communication predictors. On the left, the coupling patterns are shown on the brain and are quite different across frequency bands. Comparing the distribution with previous models, we can see MEG coupling is generally better than BOLD coupling, with significant differences between the frequency bands. And it is clear that heterogeneity and communication dynamics on the structure connectome really is fundamental to the whole picture. And with these coupling values as a reference, it's possible to do a lot of things and further explain what biological mechanisms cause these patterns. We start by comparing the coupling patterns with macro-scale cortical hierarchy. And to represent this hierarchy, we use the sensory model association axis from Sinner et al. And it is reproduced here. And we already know and have found a consistent, strong negative correlation in both. So it's time to review what our correlations look like for the MEG frequency band. So it appears the negative correlation also falls for most slower frequencies like alpha, beta, and theta bands. And there is a change in continuity from delta to gamma slowly going up until a reverse relationship for high gamma band. This is leading to the question of what might be the more fundamental cause of this preserved hierarchical relationship. With the big brain data, we are able to reference with microarchitectural regression and laminar differentiation. So big brain provides posthumous staining profile intensity values. This is a measure roughly corresponding to the cell body density, and it peaks around cortical layer 4, where there's a high density of granular cells and separate supra and infragranular layers, as reproduced here. We can see that both coupling, the correlation also peaks in such range and most similar to delta alpha and beta bands. While gamma bands, they peak in more superficial layers with high gamma band naturally correlated. And this is telling us neural oscillations and BOLD responses vary with cytoarchitectural organization on the underlying structure connections. So there are really a lot more could be further explored, but because of time limit, I'll just wrap up with some take-home messages from this project. First, it is really worth looking into the MEG coupling because it's quite different from what we had with BOLD signals and even with simpler models. Second, we again confirm structure-function coupling is heterogeneous across the cortex, and to associate with faster dynamics from MEG, the communication models really helped a lot. It could be BOLD and MEG connectomes are capturing different communication protocols across space and time. And thirdly, with many updated biological annotations, we can better understand the underlying principle. Finally, I'd like to thank my supervisor, Bratislav, and all members of the NetNeuro Lab. I encourage you to check our GitHub page where you could find data and code for this project, as well as many useful network neuroscience packages. And I'm glad to take any questions. I have no disclosures for this project.